Genetic engineering, it is the techniques of changing the genetic structure of the living cells or the individuals through removing some genes or hybrid a new molecule of DNA or altering it to enable the cell or the organism to gain the desirable traits. The following are the main needs for the techniques of genetic engineering. In first, a method of cutting down the molecule of DNA which carry the required gene to transfer by one of the restriction enzymes. So, cutting down the part of DNA desired molecule by what? By restriction enzyme, like a scissor to count the part of genetic material which is desired or wanted. A suitable carrier needed to transport from one organism to another. You may remember from first page we said DNA has some abilities or genetic material, so it is used in genetic engineering. One of them is DNA, which can be transported from one organism to another. So how transported from one organism to another? First, we need to cut it down from the first organism by a restriction enzyme. Then we need a carrier to be transported from first organism to the second. This takes place through the unit DNA carrier with the help of DNA ligase enzyme, we remember, which stick the pieces of DNA one to another. So the hybrid piece, the piece of DNA which is transported from one organism to another, this R means recombinant DNA, marked by ability to replication inside the recipient cell. A method to enter the hybrid piece of DNA, including the carrier piece for the gene that is going to be transferred to the recipient cell. So how this piece of genetic material will be transported and be a part of the genetic material in the second organism, like lysogenic cycle of reproduction in viruses. And for a method of finding the recipient cell and its generations which carry the desirable hybrid piece and separate it from the rest of the colony's members who does not contain that piece. So as we cut a piece of genetic material from one bacteria A, we want to transport B. We cut it from A by restriction enzyme. We get a virus, put all genetic material in virus, and there is colony group of bacteria B. This virus transport all desired part of genetic material to this colony, but not all of them. Some of them be affected. So in the step 4, we separate the bacteria which carry this piece of genetic material which is recombinant DNA. We separate it from the others. The following is a summary of some these needs. How we said the restriction enzyme, like a scissor. These are the bacterial proteins which are used to know the certain succession of nitrogenous bases for the DNA, the molecule and its cutting down. Also, these enzymes break the internal phosphate bonds for the DNA. So restriction enzymes, the vector DNA, and cut it down as what desired part. Sticky ends, separate it to what connect to another piece of genetic material. So we call it, because this piece of DNA is changed, not as before, new type, we call it recombinant DNA, as conjugation in bacteria which genetically changed after the conjugation. The carrier has a main role in this technique, and they are the followings. Bacteriophage Phage are simple in structure things which affect the bacteria or remains inactive. The carrier has a main role in this technique, and they are the following. First, bacteriophage. Bacteriophage, we remember, it's a virus spacecraft shape with piece of genetic material. It contains single or double DNA strip, or it may contain one piece of RNA. An example for this is the phage lambda, one example for bacteriophage. Second, the plasmid. It's a small circular form of additional DNA molecule which present in many bacteria. We remember bacterial DNA has its own genetic material, but in addition has some other pieces of genetic material which we can call them as plasmid. It carries a small number of genes which are responsible for some traits like bacterial resistance for the antibiotics. About plasmid, it replicates itself independently from the bacterial chromosomes. It can be transferred from one bacterium to another, as we studied in the conjugation of bacteria, such as yeast or fungus or some other insects. So we said in genetic engineering, in second step, we need what? We need a carrier. So this carrier can be bacteriophage or plasmid. The probe and finding desirable gene. Probe is the molecule of DNA or RNA which is monochain, and it is marked by radioactive phosphor complement for the DNA desirable succession, and it's required to find it. It means it is the original DNA, for example, this gene, which we want to cut by restriction enzyme, but probe, we know its codes. Like we need to cut the genes which help producing insulin from human. We know the insulin a protein, we know amino acids, so we can predict the codes of messenger RNA, then the DNA. And if you remember, we said the genetic material DNA can be copied by a PCR method in laboratory. So this probe we left into the cytoplasm or in nucleoplasm, it finds its complementary or with the part of genetic material with the same code. And because it's radioactive phosphorus, it can be observed easily. It's required to find it as a result of connection between the probe with the complementary DNA piece will form the double radioactive spiral. As for the insulin gene, given the probe is regarded as the RNA for this gene, usually the specialist transfer the DNA from the restructured bacteria. Then by restriction enzyme, we cut this piece of DNA by a carrier transport to what? Restructured bacteria, into a bacteria. Then the bacteria into filter paper for the purpose of finding out the bacteria contains the desirable gene. Because we radioactivate it when we transport to the bacteria by a bacteriophage, so we can observe which kind of bacteria contain this gene which is transported by bacteriophage, and we can separate it from others. We remember this step 4. When the bacteria is viewed under the UV light rays or when it is exposed to photographic film, the clone cells will receive this piece of genetic material which carries desirable DNA, and distinguished by one probe stuck to it, it will become light and shiny. So then this bacteria can divide by biphenyl fission, increase their number, and we get a colony of bacteria which contain the gene responsible for production of insulin. And it's summarized in this picture like human cell, part of DNA. We find it by a probe, cut it by restriction enzyme, then we transport it to the bacteria cell by what? By a carrier. So the new genetic material of bacteria is recombinant. Why recombinant? Because it's contained the insulin gene that through binary fission, asexual reproduction, bacteria produce number of bacteria, and they start to produce insulin, protein, or hormone. Genetic practice and what field genetics can be used to determine the succession of complete nucleolized human genome to know the genetical map, which gene in human responsible for which trait. It used in justice field, so from a nail or a cell or a hair, they identify the owner of the species. It's used in the field of following the human immigration and some organism from its environment, especially the ones threatened by extinction. Producing human insulin hormone, as we study an example for genetic engineering. Protein interferon material, clotting factors, blood proteins, and different vaccination can be used, transferring to the trait of nitrogen installation to other types of bacteria. Transferring the trait of root buds formation in the beans plant to other plants which are economically important, which now we call them, they may do many changes in living things, we call them genetically modified organisms. 7, developing researches of using bacteria in research field about the process of purification and concentration of the minerals in the soil. Also, they can be used in good ways. 7 or 8, developing the ability of microorganisms to limit some danger of pollution, which we call it bioremediation. Bacteria use it to break down the waste materials in environment, which their decay take long time. 9, applying the selection system in marriage of disdains of cows, sheep, horse, chicken, fish, and others for especially more milk or more flesh. Applying genetic concepts such as artificial selection and hybridization and regular birth to produce useful plants for humans in big amounts or maybe more better quality. Also take the advantage of twins phenomenon in cows, sheep, and to produce useful animals for human.